It's a great honor to invite Dr. Andy Gordon, Professor of Movement Science and Neuroscience and Education, the Department of Biobehavioral Sciences at Teachers College. Andy also directs the Center for CP Research. Andy has worked with Kathleen since 2008, and what started out as a mentorship on her K Award actually flourished into a lifelong collaboration. Together, they've been really kind of unraveling some of the factors that might predict or promote recovery in children with CP. And so the prompt we sent Kathleen about you, Andy, was without Andy, I might never have, ... She says, without Andy, I might have never transitioned to clinical research. Andy was incredibly welcoming of me as I learned how to apply my basic science background to working with children. So welcome, Andy.
So I'm really honored to be here and to honor Kathleen and my experiences with her. So as Claudia said, we've been working together since 2008. And we've had a number of grants, either that I've mentored along with Jack and Holly, or we have done together. So in the more than 15 years that we've been working together, we've had 33 publications, peer-reviewed publications, and 4 book chapters. So I'm going to take you through each one of them. I have time for that, right? No. I just want to say that, by any metric, this rate of productivity would be outstanding. This is just a portion of what she's done over this period of time. So it's been absolutely amazing watching her grow in her career. I'm going to just kind of touch on a few highlights of this and kind of bridge to what Randy and Jack had and Holly had said. So basically, she was interested in transitioning to human work, and she had contacted me while a postdoc in Jack's lab. And I think I first met her at a conference at Teachers College that Jack was a speaker, and he brought some of his lab mates, and she reached out to me and indicated that her long-term goal of doing some human research. So I invited her to some of our intervention studies with kids and that are done in camp settings, to observe. Well, basically, Kathleen's version of observed, she's not passive. She immediately embeds herself into the camp and begins working with kids and coming up with good ideas, and really enjoyed herself, and the kids really enjoyed working with her, and it was just really a kind of a magical experience. So as Claudia mentioned, not only is she a proficient academic, but she is a master in Taekwondo, and basically, again, achieving her black belt. And I think one of the most fun things that she did with kids was we invited her to a couple of our camps where she basically introduced, some of the skills, as a means of developing skilled learning of tricky, difficult behaviors. And I think that was really rewarding for the children and to watch. So one of the first projects that we did, basically, there had been studies looking at the relationship between cortical spinal damage and hand function. And basically, perhaps the most simplistic way of doing this from standard MRIs, which we had over many, many previous participants before she had joined, working with us, was to take an MRI and basically to look at the cerebral peduncles through which the cortical spinal tract passes through, and simply look at the symmetry. And we can create a ratio, and then basically look at the relationship between that ratio first with hand function, which had been done previously. Basically, what we showed is that if you look at bimanual hand function as measured by a standardized test, there's a moderate relationship between basically the symmetry and that function. If you look at unimanual hand function, dexterity, there's a very strong relationship. But of course, the more important question is, would this predict the child's outcome to intensive therapy? And it turns out not at all. Okay, and that had actually been, kind of a myth in the field that some kids were just too severe, they weren't going to benefit from this sort of thing. And basically, it's not predicted by the hand function or, anatomically. And, everybody's out looking for biomarkers to see which kid would improve and which kid should get therapy, I think it's great when you can find that, all the kids would benefit from this. That's really important. Let's see. So Jack already touched upon this, and she came up with the idea to write a K award, and basically to facilitate that transition. She did this study, as Jack mentioned, in his lab and wanted to do effectively the correlate of that in humans. So basically, the bottom line relative to what we were going to do is that skill training drives motor map representations in cats. I was really excited about the idea, one, because I had followed Jack's work and I knew Jack very well, and I followed Randy's work, but I wasn't an expert in animal research. I certainly got the main points, but not the nuances, and she was able to help translate those important details to what we should do with humans. So, as Jack and Holly mentioned, we did a study where we took 21 kids who received what's called hand-on bimanual intensive training, which is bimanual training done very intensively with skill training, basically skill progression, making tasks embedded into play more and more difficult and challenging the children as we go along. And then we wanted to compare that to, well, what happens if you just give them tasks that require two hands? Holly mentioned this, unskilled training, we call this, play. Basically, this had the kids play. And this was done in two separate groups. They were, the people working with the children were blinded to the existence of the other group. The people working with the play group were told, don't do therapy, don't make it more difficult. This is, they get lots of therapy in school. This is a recreational camp. Okay, and we had supervisors making sure that the individuals adhere to that rule. So what was kind of interesting, I thought, is that if you take standard measures of bimanual training, so basically a higher score in this case means better bimanual proficiency, and unimanual training, so a lower score, this is time, means better dexterity. Both groups actually approve approximately equally. So that didn't really support our hypothesis. But it didn't stop there because, and I think this is an important part that the field needs to take into consideration, is that you can't just use these clinical measures to make inferences about what's happening. You need to go into much more depth. So one is, I should just say that the kids in the skilled training group, they had goals. All the kids had goals identified by their parents and them. And the kids in the skilled training group practiced those goals because that's a form of skilled training or learning. And the other group didn't. Turns out just by using the hand intensively for approximately 90 hours over 3 weeks, both groups improved. But importantly, the ones that practiced goals improved more on those goals. Not terribly surprising. We were also interested in kinematics. And basically, this is a simple task taken from animal studies by Mario Wiesendanger in Switzerland, where simply, in this case, humans open a drawer and have to manipulate the contents inside. So it's a bimanual task. And basically, we track the biomechanics of the arms as this happens. So what happens is, when you have unilateral impairments, you tend to use your trunk more because you have a difficult time extending your arm, and you have reduced dexterity. So the group that received the play didn't have any change in how much the trunk contributes to this task. Those who basically had skill progression, basically they reduced the amount of trunk motion when reaching towards the drawer, and conversely, basically increase the active extension of the elbow during the task. So, and there's lots and lots of examples I can give you about prehension and many other tasks that kind of demonstrate this difference between this and skill and non-skill training, or bimanual and unilateral training. This is something that Kathleen was, adamant that we think beyond these kind of gross, descriptions. I think that was very important. So basically, biomechanics provides, shows differences that you can't see with clinical measurements and demonstrating the benefits of this skill progressions in humans. So along with working with Holly, she wanted to know, well, is there a neural correlate of these changes that you see with skill progression, just as you see in animal models, especially Jack's most recent work with her. So basically, she used TMS, transcranial magnetic stimulation, to map the cortex. And what she showed is, so the top group on the left is before the intervention. This is the skill group immediately after and 6 months post. And what you can see is that there's an enlargement of the sites that represent the hand, basically when they're stimulated after training. And that's maintained for the 6 months that we follow the kids. And also you can see red means that you get a more active or stronger response in the affected hand from the stimulation. So basically, presumably meaning that stronger connectivity. The unstructured play group, no systematic change. And across all the kids that we tested, the same thing. We get stronger changes in the motor evoked potentials and increase in the size of that motor map, showing the importance or neural basis of these changes. Okay, so around the time that she joined the lab, there had been a study that, we kind of joke about it as a cold wind coming from the east. It was a study where a group of Germans conducted a small study in constraint-induced therapy. And they basically, so Jack talked about, kind of this abnormal organization, a different organization where one hemisphere, the non-lesion hemisphere, controls both hands, and the lesion hemisphere doesn't have any connections. Well, they basically produced some results that said, well, kids that have this kind of organization, they don't improve from constraint therapy. We didn't have any evidence to the contrary, but because at that point we had not been using TMS, but she said, well, look at your kids. You got lots of kids with mirror movements. You got some pretty severe kids. We know that this type of organization leads to greater hand impairments. So in theory, that means that, and everybody improved. So in theory, that means that that's not true. So as part of the first R01 that we did together, we were looking at if depending upon the cortical spinal tract organization, whether kids would improve regardless of whether they had a contralateral normal pattern or this new altered pattern. And second, would that differ depending upon basically bimanual therapy versus constraint therapy, unimanual versus bimanual training? So this is the unimanual test. This is the 0, meaning that there'll be no change following training. On the left is the constraint therapy, on the right is the bimanual training. And then basically you can see that they're differentiated whether they have normal contralateral projections to the affected hand, ipsilateral, as Jack had mentioned, one side controlling both hands, or in fact this kind of hybrid where both hemispheres basically are controlling both hands. And basically what we found is in fact nothing. Both groups improve constraint therapy, bimanual training, irrespective of the organization. Okay, so basically all kids improve equally. And that was also true in relationship to the unimanual training and unimanual dexterity. So the next question for our renewal was, and this is still in progress, based upon these results, what happens if you target the hemisphere, regardless of whether it is what we think normally would be the contralateral hemisphere, or if they have this ipsilateral hemisphere, let's make use of it and stimulate that instead. So basically the idea is that, we know that TDCS in stroke has been shown, to be effective in many cases, and it has the potential to enhance motor learning. There are far fewer results, basically, in individuals with cerebral palsy. This was a study, a systematic review that we did with one of our graduate students. Basically, so for the renewal, she came up with the idea of either targeting in individuals who maintain the normal contralateral projections, either targeting the affected hemisphere, or if they didn't maintain it, basically inhibiting, or in this case, inhibiting the contralateral hemisphere. So they could receive one or the other type of stimulation. And then the same thing for kids who only have one side connecting to both. You either can excite it or inhibit it and see if there is a differential effect based upon the type of organization and the type of stimulation. So this is an ongoing study that she was tireless in, for the last year, knowing that she was going to basically take a disability and leave, and to work so hard to have our lab be able to continue in her absence. And well, it's not in your absence because you're always in our head. So I just want to point out that this was by no means done alone. We had a wonderful group of collaborators that we have benefited, and maybe more importantly, they have benefited from working with Kathleen. We have also had a large number of research assistants that Kathleen has mentored. And then I think we were lucky to have some fantastic students and postdocs, and I think they were super lucky to have been mentored by Kathleen and myself, especially Kathleen, and basically, they're all going on to, achieve great things. And I think perhaps way more important than 33 papers is the seeds that you plant for, the next generation. And, you should be so proud of, what you've accomplished in working with these individuals. So I just want to, thank you for everything that you've done. And you've been such a wonderful collaborator. You've taught me so much. I think as Randy said, you have to question who the mentor is. You were the mentor in the end. Absolutely true. And, besides the transition, I just learned so much from you. You taught me, to be a better scientist. I think hopefully a better collaborator. And certainly, I've really appreciated our friendship, throughout this whole thing. So thank you, Kathleen. Oh, you've been wonderful. Yeah, I can't wait to spend more time. Okay. Thank you.